All right, thank you everyone for joining us today for our extractables and leachables webinar. Specifically today, our topic is going to be value of data collection, collaboration, and interpretation of extractables and leachables. Really looking at, are we doing the right thing? Next slide. So today's webinar will be recorded. Slides will be available via email. I'll send those out after the presentation today, and the recording should be available on the website within 24 hours. Next slide. We will do question and answer at the end of the webinar today. In order to submit your questions, there should be a bar along the bottom that has Q&A on there. If you click on that box, then a dialog box should open up. You should be able to enter your questions in there. You will be able to see questions that other participants have entered as well. So, and then you will be able to upvote those. So if we have really popular questions, we'll try and get to those first. If we're not able to get to any questions during the webinar today, we will go through the questions and Murad will be able to answer those, and we'll send out those answers afterwards as well. Next slide. So our presenter today is Dr. Murad Rahi. He is the Senior Director of Analytical Services at APS. He's been with APS since 2015. Previous experience includes Boston Scientific, University of Minnesota, Pace Analytical, Covance, among many others. He does have several patents in drug formulations and medical devices. He has over 20 years of experience specifically with extractables and leachables, and over 30 years of experience in the analytical chemistry field. So with that, I will hand it over to Murad for the rest of the presentation.
Thank you, Monica. Hello, everyone, and welcome to this webinar. This is a presentation summary. As you can see, we're going to be talking about a lot of things. We're going to start with a general introduction to chemical characterization, issue with sample preparation and data collection, NVR for small devices, water extract, value of GCMS analysis, hexane extract, value of LCMS analysis. We're going to be talking about contamination and matrix effect on identification and quantification. We're going to be talking about detection and identification challenge, crisis, whatever you want to call it, and how it works with the libraries. We're going to be talking about data collaboration, how we spot false data from correct data. And basically, this is the essence of our presentation. Extractables and leachables at APS, some takeaways, and then we'll answer questions as you might have. I hope during this presentation, I will be able to show that chemical characterization is a true art and science that must go hand in hand with automation, compliance, artificial intelligence. Let's start with chemical characterization. The main purpose of chemical characterization for extractables and leachables from chemical perspective is to identify and quantify toxics and mutagenics to which patients could be exposed. Number two, what is the effect on the drug? As you may all know, extractables and leachable is kind of a young discipline for medical devices, but it has been around for a long time, and it takes time to improve and mature. It is basically driven by guidelines. The concept and spirit of the guidelines are very good, but the application and results, they swing from acceptable to not so acceptable. And believe me, we have witnessed a wide variation from one lab to another lab. The reason for all of that is ambiguity of the guidelines, complexity of medical devices, lack of expertise training, and lack of understanding even amongst a number of key holders. Now, let's start with sample preparation. This is where it all starts. To perform an exhaustive extraction per ISO 10993 part 12, you need to use three different solvents, or at least three different solvents are recommended with different polarity: polar, semipolar, and non-polar. You perform a sequential extraction and show extractions that the extraction is over 90% complete. How? By NVR. This is something that FDA really likes, but we're going to talk about that, how it is a problem for small devices. FDA expects no selective analysis, which means every extract should be it should be analyzed by all the platform. So, and then you write a report, submit it to toxicological risk assessment, and everything will be taken care of, supposedly. Why FDA loves NVR, and why FDA does not like selective analysis. Metal elements are analyzed by ICP-MS, and they are only available in saline. Why? Because they are soluble in water and in saline. We are not analyzed in IPA or hexane because of their solubility. So there is affinity of these compounds for this polar solvent. Now, can't we apply the same principle of solubility, like dissolves like, and perform selective analysis on other extracts like the polar and the hexane? So I'm gonna show you what I mean. Before we talk about the selective solvent analysis, let's talk first about NVR for small devices. Issue number one is determination of NVR following USP. It requires a certain volume, greater than 50 mil, surface area 60 centimeters square, and a difference between sample extract and the blank about 15 milligram or less. Issue number two is the balance. Most analytical laboratories have 5-place balances. Operating range, typical operating range, 20 milligram. So how small medical device is a problem in here? Example, we have stents, ocular lenses, dental filling, mesh, etc. Those devices, they weigh in between 10 and 100 milligram. Let's say we use a device with 100 milligram and extract, and suppose we extract 1% of the 100 milligram in the first extract. In the second extract, let's say we get 50%. This will be equivalent to about 0.5 milligram. Now you do the math. How many devices are needed to comply with the USP requirement with the balance and with NVR? 30, 50, 100, 5,000? I've seen as many as 5,000. Given that you have to run triplicate analysis on 3 different solvents. Another issue is evaporation of volatiles and semi-volatiles. You're heating the solvent at 105 degrees. So how can we account for those volatiles and semi-volatiles? The alternative and the good solution for NVR, especially for small devices, is chromatography. There will be no problem with the USP. There will be no problem with volatiles, and save a lot of devices and a lot of cost. And anyway, and this is the funny thing, quantification and identification of extractables and leachable is based on chromatography. So ISO 10993, as well as USP, they recommend chromatography, but they prefer also NVR. This is where the ambiguity in the guidelines comes. And when you don't do NVR for FDA, they question why the NVR is not done. Well, give me a break. How do you do NVR for such a small compounds, for such a small devices, and yet use the balance? What if 5,000 devices? Well, that's a problem. This is why I have issues with this kind of analysis and requirement for small devices. Let's go now back to the principle of solvent selectivity, or like dissolves like. What do we mean like dissolves like? Everybody knows that from high school probably, that polar solvent dissolves polar compounds, and polar compounds are water soluble. Non-polar solvents dissolves non-polar compounds, and those are analyzed by GCMS, as well as polar compounds are analyzed by LCMS. This is why we have LCMS. The semi-polar compounds, the IPA, you can see it bridges a little bit between the polar and the non-polar. Well, look, polarity increases in this direction, non-polarity increases in this direction. So the non-polar compounds, which are soluble in hexane, there is no way these compounds are soluble in water or detectable by GCMS. And since we have a buffer zone in here by IPA, and IPA is suitable for analysis by GCMS as well as by LCMS, then what is the benefit of taking the hexane extract, analyze them by LCMS, or the water extract and analyze them by GCMS? So let's go to problems with water extract using GCMS analysis. Okay, now direct inject of water extract is incompatible with GCMS. You know, GCMS is under vacuum and it has no air, even air leak. So how can you inject water? It will affect the column, it will affect the source, and so forth. Okay, we do headspace analysis. Headspace analysis on water extract is limited to volatile material. You cannot boil water more than 100 degrees because you don't want to saturate the headspace with vapor. Now, a third option is do liquid-liquid extraction. Fantastic. This is what FDA recommends. Do liquid-liquid extraction at different pH. Oh, what we use in here, non-miscible solvent like hexane or DCM. Even if you use ethyl acetate, it's not suitable because ethyl acetate will hydrolyze at high pH or at low pH. So you have to be careful which solvent do you use. So we are limited to either hexane or something like DCM. Okay, let's evaluate the value of this re-extraction at different pH as FDA recommends. Let's go to the second slide here. What do we have in water? We said polar compounds are soluble in water. Ionic compounds soluble in water. At low pH, what we extract? We extract polar compound, acidic compound, which have proton donors. This class of compounds are typical candidate for electrospray in negative mode. Typical candidate. For high pH, what do we extract? There are polar compounds also. They have basic groups like amine or something like that. And they are proton acceptor. And those are typical candidate for electrospray positive. Both type of compounds at low and high pH are poor candidate for GC. That is why LCMS is for ionizable compounds which have great affinity to water. These compounds often require derivatization to make them amenable for GC. Now, derivatization is mostly performed in aprotic solvent. What is aprotic solvent? That doesn't have proton donor, like water, methanol, ethanol, and so forth. And then it requires evaporation of the water, reconstituting into a protic solvent, such probably acetonitrile, and then do the derivatization. Well, it's not an easy process. It takes a lot of time, and it may create a lot of problems with the extract. So we'll talk about this one a little bit more about the matrix effect. Now, what is left is the neutral pH. Neutral pH, mostly they extract nonpolar and semipolar. The nonpolar already extracted in hexane and analyzed by GCMS. The semipolar extracted with IPA and analyzed with GCMS and LCMS. So is there any value for this extraction? The answer is yes, only if the device is extracted with water. Only if the device is extracted with water. And this is the school of the EPA when we didn't have LCMS. We take the water, we extract it with hexane, we extract it with this and with that, and then we have to run them on GC. But with the use of LCMS, I don't see a good reason for this re-extraction at different pH. Or it is used for conformation, additional conformation of a particular compound. So for this reason, we can pass on to the second part of the issues is hexane extract. Same thing, direct inject, incompatible with aqueous mobile phase. Oh, let's do evaporation and reconstitution. In what? Water, acetonitrile, IPA, because they are compatible with LC? Fine. Remember, hexane is a good solvent for lubricant, machine oil, wax, hydrocarbon, a lot of polymers, adhesives, and so on. They risk the formation of particulates when you put them into acetonitrile and water and IPA to redissolve them. Those things may not be soluble and they start forming either a precipitate or particulates or clusters, or they have phase separation. They can contribute a lot to instrument contamination. Contaminants are really hard to remove from LCMS, especially this type of contamination. This is why I do not favor injecting hexane extract on LCMS without further cleanup, which is not allowed for device extract because you cannot clean the device extract. This will be allowed only if you are doing targeted analysis. Specifically, you are looking for something, then you can clean the matrix. The third option is one-to-one dilution as recommended by FDA. We still have the same problem as above, you see? So there is a really big problems with hexane to LCMS. Now, let's go back to see how identification and the quantification affect instrument sensitivity and so forth. You know, most laboratories complain about lab contamination, interference, ghost peaks. Is it lab contamination, instrument sample handling, and so forth? Or is it matrix effect? Quantification and identification are not only affected by contamination, they are also affected by what I call matrix effect. So what is matrix effect? Device extract depend on device contamination, on device construction material, manufacturing process, sample preparation, and so forth. They may contain a lot of organic and inorganic ions. Ions including metal ions. And metal ion is a really big problem for LCMS. So maybe that's why I always advise to run the water extract first on ICP-MS to see what level the metals in the extract before putting those on the LC. It may not be a bad policy if you guys are in the lab and you are listening. Ions have a great affinity to form adducts, cluster, complexes. They present direct effect on quantification, identification. They could affect, the effect could be like signal suppression or signal enhancement. It does not affect all extractables the same way. Each extractable is basically affected differently because it depends on its structure, on its polarity, affinity, and so forth. They may have same mass to charge ratio as target analyte and interfere with identification. They can have direct effect on chromatography, such as peak shape and elution pattern. So what else? Instrument parameters affect quantification and identification. How? Column, voltage, flow rate, etc. Mobile phase. A slight change in mobile phase have a tremendous effect on the mass spectra. Now here is a reference for you about tips and tricks on how to deal with contamination. As I said before, some of the contamination, some of those tips and tricks may not be applicable because we cannot clean the matrix before we analyze it. So you can see how complex and challenging extractables and leachables. It is not only limited to matrix effect and contamination. You know, a good number of key holders think that extractables and leachable identify every compound in the extract. A misunderstanding. I hear this all the time. Let me tell you, lots of extractables go undetected by GC, by LC, by UV, and so forth. You know, I wonder if not too much time and resources are spent on trivial analysis and FDA audits. FDA audits like hexane by LCMS, water by GCMS, 90% saline with ethanol for FDA is not polar enough. Demonstration of stability of extract, if not analyzed within 24 hours. Well, give me a break. The extraction lasted for several days at high temperature. Am I worried about stability at room temperature for an hour or for a couple more days? Or extraction for 72 hours following the guideline may result in saturation and false determination of NVR. This is one of FDA audits. And yet, it is of no concern for biocompatibility testing. I believe the safety of the patient would be better served by performing additional selective analyses on potential compounds that they go undetected and ensure that the device is free of non-extractable toxics. And this is what I call detection crisis in my summary, present in the first page. So what do I mean detection crisis? Let me tell you, as I said, lots of compounds go undetected, even toxic ones. How? They may degrade during extraction, polymerize during injection, form adducts hard to identify, pass through undetected because they require selective methods, selective techniques, different instrumentation for detection, or simply they are not extractables. What do I mean not extractables? Here is an example. I'm going to show you a simple example so you can understand what I'm saying. Let's suppose we have a heart valve constructed of collagen cross-linked with an epoxide or glyceraldehyde. it failed biocompatibility. They come and to ask for extractables and leachables to identify the toxic material. ENL does not identify anything. Where is the problem? What happened? The problem is what I call residuals bonded cross-linker. So this is a typical example. We're going to use this cross-linker in here, which is an epoxide. And it can have different groups, 2, 3, 4, whatever, that we use for cross-linking. As the polymerization or cross-linking reaction goes on, so you can see the amino group attack the epoxide on here, and it attacks in here. It forms this, it starts the polymerization, and this polymerization goes on to form oligomers, long chain molecules, and so forth. And then the cross-linker, we wanted to cross-link this molecule now. So we add the epoxide, and it starts to cross-linking. Now, what will happen in here, some of the active groups, they get trapped within the molecule, either because of steric hindrance, they cannot react anymore, or because the reaction stopped soon, or the curing of the final product was not good enough. So those, they still on the device, they are not available to be extracted. And then when the device is used for biocompatibility testing, they bloom to the surface, or they are already on the surface, and they cause the problem. So you can see what I mean by non-detectable toxic materials. Now, let's go to how contamination can affect the analysis. This is a very simple exercise, a mixed reference standard of nylon monomer and antioxidant additives. You check the chromatogram A, so you can see the signal barely above the baseline. And you can see this pump, which is a contamination. Now, for a good chemist, he knows that this is not acceptable. So there he goes and he cleans the instruments, the clone, the injector, the whatever, and then he gets this better detection, better signal. And then he can go and also optimize the instrument parameters, and he gets this chromatogram. Now you can see the difference between this chromatogram, this one, and this one. Now, for untrained chemist, he might use to run the samples using these conditions. For a trained sample, he wants to use these conditions because he knows better. And then you can see the difference, how much difference the results would be had we used this condition, that condition, or that condition. So there will be a direct effect on the quantification and identification, as we will see later on those samples. Now, let me show you a simple additive effect, how it affects the analysis. This compound is analyzed with no additive. And you can see there is no, the molecular weight is 320. Basically, there is no 320 over here, no molecular weight. You add ammonium fluoride and look, the molecular weights, how high it is. In addition, it forms an adduct, 339. And then look at the spectrum right here, the difference between here and right here. So now we have an adduct. You have two different spectra for the same molecule. How the library is going to differentiate between this one and this one? We will talk about this a lot more in a second. So let's go in here. This is a compound that is analyzed at 10 volt cone voltage, optimized for that. Well, believe me, if you change the voltage a little bit, you may get completely different spectra. So at 10 volt, you see there is no detection whatsoever for the molecular ion. But if we add, if we use ammonium acetate So this is the mobile phase, 3 millimole ammonium acetate. Look at the pKa, pKa 476. Now, if I change the mobile phase from 2 millimolar ammonium acetate to 2 millimolar ammonium bicarbonate, now the difference here you can see pKa 76, pKa 635. Look the difference in here. No molecular ion, huge molecular ion. And then what you see also, an adduct, 2M-1, which is 2 molecules coming out together. Is this an adduct, or it is like coelution of enantiomers? Enantiomers, because of different configuration of the molecule, it has an asymmetric carbon in here, so it can exist as R and S for those people who know a little bit of about enantiomeric chemistry. And then maybe they are not separated, they coeluded together, and this peak is not an adduct really, it might be coelution of the two together, coming together. So in any case, what if we, if the extract of the medical device contain not ammonium acetate, but let's say ammonium oxalate, ammonium chloride, ammonium sulfate, ammonium phosphate. So you can see it is going to be completely different spectra for each adduct we have. Let's talk about this one. I like this one. This is an extract, actually an extract. This is a spiking of a standard by using a mixture of different compounds for GC. 5 microgram injected in the control. So you can see the peaks, very nice shape, separated, no problems. We use those to perform system suitability. So what was done is we take the same mixture and we spiked it in a 72 extract, in 96 extract, and 120 extract. Look at those peaks. This is naphthalene. You see, basically unaffected. It gets a little affected and it goes okay. But if you look at 1,2,4-dichlorobenzene, this is a little bit more polar than that one, okay, because of 1,2,1,4-dichloro. Look at it here. In the second extract, it is gone. In the third extract, it is gone. It goes back to being detected in the last one. The same thing. If we look at this one, same thing. So what is happening here is when we do the first extract at 72 hours, there are, let's say, as we said, adducts, ion, lots of ingredient in that extract. And those, they affect the detection of the standards, not only the standards of all the molecules, because every molecule is going to behave differently. It depends on its affinity, on its polarity, chemical structures, and so on. So the effect is not homogeneous across the line. It will be, because in the extract, we don't know what we have. It could be acidic, it could be basic, it could be neutral, and so on. The neutral molecule, naphthalene, wasn't really affected that much, but a little bit of polar compounds gets really affected. So what if we are doing the same thing? If we have like phenol, acidic, basic, so the effect is really tremendous. Now, is this behavior always detected or exhibited? No, because sometimes we may have, it depends on what is extracted in the first, second, or third. So let's go to the third one. We talk a lot about relative response factor. You know, this is from 1093 part 18. And all the, we determine the analytical evaluation threshold, quantify above it, forget about it below it, and so forth. And then that's what I wanted to make the point is how it is going to be affected, how it's going to be affected by the matrix. We cannot take it for granted. So I know the new revision recommend different different standards to elute a different profile. And this is how we do it at APS. And then every groups will be used for the closest standard. But still, this is really too much theoretical because we don't know how it is affecting, how the matrix is affecting the internal standard. Those pretty much like theoretical. A lot of, I say a lot of conferences and publication talking about relative response factor varying from 100 to 400. Yes, if every molecule has its own response. But what if we do a study on the matrix? We can't because we don't know what's in the matrix. It depends from one device to another device, from one extract to another extract, a little bit of additive here, a little bit additive. This is a huge problem. And at times we do our best. So see, I'm running really quick. Here is a webinar. Here is a webinar that was addressed by the manager of library for Thermo Fisher. And he called it identity crisis of small molecule. Why small molecule? Because a small molecule, this is what we look for in extractables and leachables. And we don't look for large molecule because they are not gonna be extracted. Now, and he already acknowledged crisis in identification of small molecule. He's the manager of the library. So let's see what we can learn from this. This is a typical library that people have in their laboratory. For GCMS, so we have the NIST and the widely most commonly used for GCMS, and then for LCMS, basically everybody has his own database. For us, we use the MZ Cloud because we use the Thermo Fisher most of the time. So how we do database? Usually, you use a reference compound, you inject it, you collect spectra, and then you put it into the database. It is easy for GC because the collection is done all the time at 70 electrovolt. But for LCMS, there will be multiple spectra depending on the mobile phase, depending on the adducts, depending on this, depending on that. It is an enormous task. So what we do what we call in silico spectra, which is computationally generated spectra based on algorithms, bond dissociation approach, and match to experimentally obtained spectra. What if the reference is not available? And most of the time we don't have references for extractables and leachables. And as I said, what about the adducts? What about the small variation? Another problem with libraries is they contain similar in silico spectra. Database hosts millions of structurally similar compounds. Believe me, what I did, I did a query for C22H2009. Why this one? Because I took this compound just by curiosity, and I did its empirical formula, and I found out it was C22H2009, molecular weight 428.4 gram per mole. Then I did a query on PubChem to see how many compounds this formula has. Believe me, this is what it is. I have the link for you to go and check it out. I got 168 compounds with the same empirical formula. So I picked a few of those. You see, like this one is a cross-linker, and you can see here, we have an adduct here, an adduct here, another adduct here. Let's look over here. It is trihydroxybenzoic acid. The same thing in here. It's with another different adduct. And look at here with another glycolate. Over here, a crown ether. Those are representative of the 168, and most of those compounds, like this one, the phthalate in here, typical compounds we found in devices. So you can see how we're going to be able to identify by the library which is which if the library gives me a molecular formula C22H2009, which one is the right one. among all the possibilities. And now I didn't really consider the effect of the adducts, the effect of this, the effect of that. Those are like simple molecules, it's just simple, straight molecule, not in a matrix. So you can see the complexity of this one. If I want to go over here, I can show you the clusters and adducts formation, potential formation. And this is a reference for you to read about potential contaminants and how to detect them in laboratories and in while you are running extractables and leachables, even not extractables and leachables, anything else. So you can see typical adducts, proton, ammonium, sodium. Here it is solvent adducts, acetonitrile and methanol. Here it is aldehyde and water for polyethylene glycol. You see so you can see phthalate. You see the phthalate with acetonitrile, phthalate with sodium, phthalate with solvent, triton, clusters, all kind of clusters. All those ions, they have the affinity to form complexes, add together. Now you're going to tell me, some of the people, they're going to ask me, hey, Murad, you know, high resolution mass spec should take care of adducts. I can tell you, yes, if you are doing target analysis, lots of adducts, when you do general screening, they don't get sorted out unless you do target analysis for targeted compounds. So a lot of those adducts can get through. And the formation of these adducts, they can give you same MZ as your analyte. You know, I routinely go and work as a consultant for laboratories, and I review a lot of data. Running out of time, but we have to do a little bit fast. So this is the data, typical data, collected on GCMS. The extraction is methanol. Look over here, heptacozone, 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 heptacozone. Give me a break. How many heptacozones there is in a mixture? Only one. Heptacozone is saturated molecule. It doesn't have multiple molecules, multiple isomers to elute at different retention time. The same thing over here. Let's move on. Here is another example, which is which is against the principle of gas chromatography. This is also a hexane extract, typical example, figure 5F, taken from a report. They identify what? They identify a compound, they call it N-dihydroxysuccinimide. So I Googled this one, I couldn't find an N-dihydroxysuccinimide. I found out N-hydroxysuccinimide, okay, with this molecular formula. And it has a melting point 95 degrees, which is really against all the principle of gas chromatography. In gas chromatography, the higher the boiling point or the lower the boiling point, the faster it elutes. Like, look at this compound, 174, 216, 253, 278. They follow the pattern of elution. But if you look at a solid to elute before a compound dodecaine with a boiling point 216, this is against the principle. And this one is very, very polar. And if it is so very polar, it is not extractable in hexane. It should be identified either in water or in IPA. Let's move on. This is another example here. The same thing. This is identified in hexane. I looked at this compound here, the way it was identified in the report. I couldn't understand what it is, so I Googled it. It is anti-neoplastic agent. Well, I looked for the structural formula. I looked at the structural formula, a very polar compound, an amine, a carboxyl hydroxy. There is no way in the world that this compound can be extracted with hexane or be in hexane. If it were in water, I would have accepted. So this is how you identify compounds. Now, let's move on. This one is a little bit, well, everyone is a little bit different. This one, it's an anhydride, and TMS. Anhydride where? In water and ethanol. Well, anhydride is not stable in water. They degrade immediately. They break down to phthalic acid. TMS is not stable in water. So when I see compounds like those in water extract, I just it kills me. I can't understand it, that we don't really teach people what to look for. So in any way, let me catch my breath a little bit. I'm getting really excited here. So why is the library? Why is the library? Because if you go back in here, you see, I forgot to mention in here. Why, if you go back in here, all these compounds were identified by what? NIST library. So all those compounds, they rely on the library, and this is what the library tells them to do. Oh, great, the library is telling me what it is, so let's go for it. So you can see you cannot rely too much on the library. Now, as the artificial intelligence, the building of the library progresses, we probably would have to include one day some kind of algorithm and say, you know, if the compound is so soluble in water and insoluble in hexane, it should not be detected by the mass spec. So some kind of algorithm probably in the future will help us take collaborate the data from GC to the LC to the UV based on their polarity, based on their solubility, and so forth. And then that library, the results would say, hey, this compound should not be identified in hexane, so it is a bad identification. Maybe the future is there. I do not know. I hope so. Maybe it is. Let's move on. Here's another compound. Here's another compound. You see the same story. They couldn't identify, they identified this compound in GC and they failed to identify it in LC when all the data shoots that this is what it is. You do a quick library search, it's all support that this is the data. They couldn't collaborate a little bit between GC and LC. Let's move on. So we can have a little bit of time for you to ask questions. So how we do things at APS? Oh, at APS, we do, I think, a good job, believe me. First, we target study design for each device. Each device is different. We conduct a pilot study for complex and biodegradable devices, and we pay a lot of attention on that. We use qualified, calibrated, sensitive instrumentations. We ensure lab instrument contamination under control. Quantification, we use mixed internal standard. We truncate the quantification. We provide training to ensure proper interpretation and collaboration based on principle of chemistry and best science, how to interpret mass spec, UV, GC. We use commercial library and we do have our own library. We continue to expand in-house library. We ensure spectrum is compatible with library identification. We question unexpected results. We provide recommendation and root cause finding. We maintain good relationship amongst all key holders. And finally, this is the sad story. We follow FDA requirement, analyze extract on all platforms to ensure the box is checked. So for our APS, we do have a GCMS headspace for volatiles, GCMS for volatiles, semi-volatiles, high resolution mass spectrometry for semi-volatiles, non-volatiles, positive and negative mode, ICP-MS for elemental analysis. We do a lot of target analysis because, as I said, we do target analysis when we feel it is necessary to see if we have unidentified compounds. So where are the takeaways? Quickly. We do emphasis on data quality as much as process and compliance. You don't believe me that ISO certification compliance is no guarantee for data quality? Check this presentation. It was presented at SOT 2018. 4 different laboratories couldn't, for simple exercise, could not provide similar data. Search library artificial intelligence is a tool, not the tool. Toxicological risk assessment, as good as the reported data. Data, as good as the one behind the machine. Selective analysis, whenever required to add value. Analysts should possess at minimum good knowledge of chemical, physical property of solvents, chemical polymers, additives, and so forth. So we have to know the chemical, we have to know the boiling point, the TG, coefficient of distribution, the melting point, all kind of things. Good understanding of chromatography and spectroscopy. What does it mean, you know, a peak eluting early or a peak eluting late? How the spectra corresponds to the peaks. They have to know how to interpret and collaborate data between GC, LC, UV. At least do a good literature review, data collaboration. So in summary, let me tell you what, you gotta think out of the box. In extractables and leachables, it is not following a process or an SOP or a chart or a form. Every design is different. If you don't think out of the box, you get a lot of confusion. Yes, ENL is a challenging multidisciplinary effort. It requires a team to speak the same language. Here at APS, we make sure we know that 10 plus 10 is 20, and 10 times 10 is 100, and 10 divided by 10 is 1. We speak the same language to ensure our utmost safety of the device. So at the end, I like to finish by thanking you so much. And I will,
All right, thank you so much, Murad. We do have a couple questions here, so I will ask these out loud. The first question we have is, does APS have toxicology support for a toxicological risk assessment of ENL data?
We contract with a toxicologist, and he works with us for a long time. He's a very good one, and we are very happy with his reports. And we discuss with him, we solve the problems together, and this is how we work the toxicological risk assessment with that individuals.
Great, thank you. So the next question is, how do you perform extraction in apolar solvents in the case of nanoparticles, nano-enabled devices, that aggregates in organic apolar solvents like hexane? And following up to that, what's the FDA recommendation in this case?
Well, this is a really good question. How you do it? You have to, at the beginning, to run a little pilot study, if not an extensive pilot study, and see which solvent is most compatible or which solvent is suitable, and then you go from out there. You cannot really design a plan before you do tentative investigation in the beginning. So basically you have to do your homework in the beginning and find out which solvent is compatible, which solvent is not compatible, run chromatography, see what you are getting, and then collect all this information and then build a plan based on this information. So there is no quick answer for complex questions like those. It always takes a lot of investigation before you do a good plan or a suitable plan.
All right, thank you very much. We've got another one here. For a known contaminant concentration, what is the process to determine if the level of contaminant is acceptable from a regulatory, safety, and other perspective?
Most of the time, if you look at the detection level of your instrumentations, contamination is acceptable if it is less than 20% of your LOQ, which is the limit of quantification. And if we are talking about the limit of detection, which is most of the time it is 3 to 5 times the LOQ, then really it is a personal judgment. At least it has to be 50% of the LOD minimum.
All right. We've got another one here. How do you avoid interference in LC-MS?
As I said, those are problems in laboratories, and there are a lot of things that they can contribute to interferences. They could be contamination from the lab, from the glassware, from handling, from packaging, from the device itself when it was manufactured, processed. Those are impurities inherited even in the raw material. How, how do you avoid them? I would imagine you do your best to identify those interferences. Once you identify what they are, what those interferences are, then you can look for the source of those interferences and take care of that. Short of knowing what are those interferences, it will be hard to get rid of them. So.
All right. We've got one more here. Oh, we just got another one. So we'll do this one first. Phthalates shows more interference from LCMS grade solvents, and the noise level, this one's pretty similar. The noise level is higher, and just kind of how to avoid that. So I think we kind of just went over that.
Can you repeat the question, please? I got distracted a little.
Yeah, sorry. It's phthalates shows more interference from LCMS grade solvents, and the noise level is higher. So that's a pretty similar question. I think we've kind of answered that one, unless you had anything to add.
Yeah, you're talking about noise level is higher
yeah,
than
noise level is higher with the phthalates.
Well, the noise level, where is the noise level comes from? We have to understand what contributes to signal to noise level. A lot of things can contribute to signal to noise level, to higher baseline, to a rise in the baseline and noise level. It would be contamination, it would be column, it could be a filter, it could be solvent, it could be a lot of things. So the first thing you gotta do is use a standard in a particular solvent and check if that standard is okay. If it is not okay, then you may have to clean your system from contamination, from change the column, change, if you were talking about cleaned, it's a matter of cleaning from, it starts from the injector all the way to the detector, passing by. It might be a connector, it might be a tubing, it might be a filter, a guard filter, or this or that. So it's a maintenance of the instruments, calibration of the instruments, and ensure that the instrument is calibrated, it's sensitive, it is suitable by checking with standards outside of the extract. So basically, that's what it is.
All right. We've got another one kind of circling back to the previous question about the nanoparticles and apolar solvents. So what is the FDA recommendation in that case, and is a lipid or protein mixture apolar enough?
Well, I would imagine if you go to the FDA 10993 and read about the nanoparticles, there is a good description of how to do nanoparticles, and it is still not that the guidance is not there, but they give you some ideas. So, when the problem presents itself like this, we try to evaluate the composition of the nanoparticles, the compatibility with the solvents, and then go from there. I don't really have a direct answer for this question.
All right. So we are getting close on time. So if anyone does have to leave, we completely understand. We'll continue to answer the questions here since Murad does have a little bit of time. So the recording will be available with all of these questions answered. So next question is 37 degrees Celsius or 50 degrees Celsius is used in extraction. Is there any concern some VOC may be lost at 50 degrees Celsius?
Oh, absolutely. This is the irony of it all. the guideline, it says you extract at 50 degrees. You know, to do exhaustive extraction, the notion of exhaustive extraction is lost. What is exhaustive extraction? We take exhaustive extraction as something like exaggerated extraction, which really it's not what it is. There is a whole lot of confusion about what is exhaustive extraction. But in any way, if we do 50 degrees, yes, there are a lot of compounds that they may get degraded. They may not survive the 50 degrees extraction, especially if you are talking about valves with tissues. Their TG is very, very small. Most of the time is less than 50, 55 degrees. And if you wanted to ensure that the device is not affected by the solvent as they require, then 50 degrees may not be applicable. So what you can do, that's why a pilot study is important, because if you see effect of the solvent on the device at 50 degrees, then you can justify it and use lower temperature.
Great, thank you. Next one we have is, what impact will the latest version of ISO 10993-18 have on chemical characterization testing? Are there any interesting changes coming in?
Yes, it is a little bit. There is more clarification with the new version, especially as it relates to how to define AET and what is and how to use multiple standards, let's say. And also they have emphasis on surface chemistry. So it is pretty much more clarification to what we had before, but I can tell you some of the clarification are not good enough. They still subject to interpretation, such as what is the DBT for a medical device, for a long-term implantable device? Is it 1.5 microgram per day? Or we can accept 120 microgram per device, given that 120 microgram per device is safe if you are taking it for up to 30 days. What if the device doesn't have more than 120 microgram? So those are like, you know, subject for interpretation, and especially FDA, they still dwell on 1.5 microgram per day. How do you do 1.5 microgram per day? You have to do a dissolution rate. And for medical device, and they talk about dissolution rate, or they call it release rate. For medical device to perform release rate, it is a very complex issue. It is not like pharmaceutical drugs where the dissolution is already established. Every device has a different size. Every device has a different configuration. So what kind of container are you gonna use? At what temperature? How are you gonna stir your bath? All of those are really important questions to answer and to define in the future, rather than say, hey, 1.5 microgram per day, even though the confusion here is, instead, a lot of people, they think 1.5 microgram is 1.5 microgram per device, and they were pushing analysts to detect 1.5 microgram per device. Let's say we have a large device with a large volume to extract. How are you gonna detect 1.5 microgram in 500 milliliter? There is no way in the universe a detection limit can be used. So you have to do concentration, you have to do this, and every time you manipulate the extract, you tend to risk contamination, to risk formation of adducts, to risk getting ghost peaks. It's a very complicated issue, but it is not, you know, we try to do our best in here and establish best science for every time we have a complex problem like that. So I hope I clarified a little bit.
All right. Next question is, how do detection limits of the test methods vary, and how important is that?
Oh, this is very, very good question. As I said it, you see, you can establish a detection limit by using a standard. But what would be the detection limit when you are really doing the extracts? This is where the detection limit should be established on the extract. And the problem becomes, if we want to do it on the extract, as we have seen in this particular example, each extract is different. So where are we going to draw the line? You see? So it's a, you know, you do your best, but that's how it is.
All right. Thank you. We've got a couple more questions here. Next one is, can you please share your experience in dealing with the matrix effect?
Experience with dealing with the matrix?
Yeah.
I don't know if there is any experience on this earth to deal with matrix effect from a medical device, as long as we cannot touch the matrix for further purification. So the only thing we can do, if we have water extract, to minimize it, most likely it has a lot of ions, and those ions, they have direct effect on the detection. So what we can do, we can divert for a certain period of time the flow to the mass spec. So this will be established based on each extraction. You can divert it, and then you can do several experiments and find out at what retention time or at what time I should start the flow to the mass spec. You cannot go for too long because you can miss a lot of analytes of interest. So this is one way of dealing or minimizing the effects of the matrix. At times, there is no way you can get rid of all of those because some of the adducts or some of those interference may elute later on. Because if you take like phthalate, as we said it, phthalate and this and that, they have late eluters. But in general, it is not a bad policy to divert the flow early on for a minute, half a minute, it depends, you know, and this becomes really something acceptable. As I said, if you do the analysis of water extract on ICP-MS, you can have a good idea about the metal content. And then you can figure out how can I get rid of the metal content before I put them on the LCMS, because metal is really bad for the LCMS at minimum. But how we do with the organic and inorganic ions, other organic and inorganic ion, that's another big problems. As I said, the only thing we can do is that. Beyond that, there is nothing you can do. If you were doing targeted analysis, then you can take the matrix, purify it, because you know exactly what you are looking for. Then there will be no problem with interference, and we should be able to get rid of the interference.
All right, we've got 2 questions left. So the next one is, for medical devices, if the device already passed biocompatibility testing with polar and non-polar solvents, is it still required by the FDA or ISO to perform ENL testing for a certain solvent? And what's the relationship between ENL and biocompatibility testing?
Oh, this is a debatable question. Everybody can debate that question. You know, the purpose of ENL, beside chemistry or chemical characterization, is to see if we can avoid using animals to prove safety. Now, if it has passed biocompatibility testing, you still have to do extractables and leachables if it is an implantable device, especially if it is a long-term implantable device.
All right, so thank you. And last question is, when MOS is less than 1 and compound ID is speculative, FDA wants more identification to reduce uncertainty. However, re-extraction and further ID require many test articles. In that case, do you recommend simulated use extraction or show extractable are below AET?
In this case, this is what simulated use extraction is for. So you submit the device to a simulated extraction and you justify it and you go from there.
All right, thank you very much. That's all the questions we have for today. As I said, the recording will be available afterwards on our website, and slides will be sent out as well. So thank you, Murad, and thank you everyone for joining us.
Thank you for you. Thanks very much.
All right, bye-bye.